Good morning everyone. I am Dimpi Singh, Assistant Professor of Mahatma Gandhi Institute of Health Informatics. Today I am going to discuss about Introduction to Biological Databases. In my previous class, I have discussed about bioinformatics. What is bioinformatics? So, bioinformatics, as I already discussed, that bioinformatics is a branch of science which is used to help out and solve out the biological problems related to biological sequences and related to biological structures like DNA, RNA and protein. So, database, it means the collection of data. What is data? Is data is similar as a information? No. There is data and informations, both are different terms. Data is a raw facts and figure that is meaningless, and information is a processed data which is analyzed data which is used for the analysis. So from data we can get the informations. So biological databases is the collection of biological data. Biological libraries like DNA libraries, RNA libraries, protein libraries from the experiments, high throughput experiment technology, computational analysis. So basically we can say that biological databases are libraries of life science informations. They contain informations from research areas including genomics, proteomics, metabolomics, microarray gene expressions and phylogenetics. Phylogenetics terms discussed about that phylogenetic is the used for evolutionary relationship between the species. And informations contained in biological databases include gene functions, structures, localizations, clinical effect of mutations, as well as similarities of biological sequences and structures. So what are the biological databases? So it is a collection of data that is structures. Data should be in structured form as a particular structures like tables, like arrays and searchable, updated periodically and cross-referenced. Stored biological data in an electronic form so that we can access the data. Purpose is the systemizations of databases, availability of biological data, analysis of computed biological data. So what are the features of biological databases? So biological databases are the high volume data containing databases. Data curations as the main feature. Data integration, it means the data from the various sources we can store in biological databases. Data sharing, with the help of biological databases we can share biological data to other organizations, and they are dynamic, and data should be dynamic in the biological databases. So based on the manner of data storage, databases are divided into three types: primary database, secondary database and composite database. So primary databases in original form, it is stored by the experimental data, by the researchers, by the scientific community. For example, GenBank and Swissprot. Secondary databases where the value added databases with derived informations from the primary databases. It means the secondary database is the database when we analyze the primary data from the primary databases and then we submitted the data in secondary databases. Composite database, combined primary databases. Redundant and non-redundant databases, more than one copy of each sequences. And what is database? Specific sequence database. So primary databases, they contains biomolecular data in its original form. As we all know that what is primary data. So the primary data is a data which is collected by the researcher and scientist. So the primary database is the collection of primary data. Experimental results are submitted directly into the databases by researchers, and the data are essentially archivals in nature. Once a database accession number is given to an kind of data in primary database, then that data cannot be changed further. So what is the meaning of accession number? Accession number is a unique number of a particular sequence and structures. So examples: GenBank, EMBL, DDBJ. EMBL, here EMBL is the European Molecular Biology Laboratory. DDBJ, it means a DNA data bank of Japan for DNA RNA sequences. SwissProt and PIR for protein sequences. So there are different type of databases. Here EMBL, GenBank and DDBJ are the nucleotide databases, and SwissProt, Tremble and PIR is the protein databases. So as I already defined, that primary databases are also called as archivals databases. They are populated with experimental derived data such as nucleotide sequences, protein sequences and macromolecular structures. Experimental results which comes from wet labs directly into the database by researchers and scientists. Then the data are essentially archival in nature, so we can access the data. Once given a database accession number, the data in primary database are never changed. They form part of the scientific records. For examples, ENA, GenBank, DDBJ, Array Express Archive and Geo, Protein Data Bank, that is PDV. Secondary database, it contains data derived from result of analyze the primary data. When we work on primary data, we analyze the primary data accordingly. For example, if I can analyze the SSR from the biological sequences, from the nucleotide sequences, so what I can do, I can access the particular data from the primary databases, and then with the help of biological tools, I can find out the SSR, simple sequence repeats, microsatellites, mutations and variations with the help of different type of tools. There are different type of tools like MEJR tools, which is a Perl scripted used to determine an identification of SSR, simple sequence repeats, which is a very important part of the sequence and has more information about that particular sequence. So the data is secondary database is either manually created or generated automatically, and it contains some valuable information such as about mutations, evolutionary relationship between sequences, between structures. So there are different type of secondary databases like ProSite, it means protein site, PFAM, it means protein family, blocks and prints. So secondary databases comprise data derived from the result of analyzing primary data. As I discussed earlier, secondary databases other draw upon information from numerous sources, including other databases, controlled vocabularies and the scientific literatures. They are highly curated, often using a complex combination or computational algorithm and manual analysis and interpretations to derive new knowledge from the public record of science. For examples, Interpro, Uniprot in variations, functions, regulations and more layered onto whole genome sequences. However, many data resources have both primary and secondary characteristics. For example, Uniprot, for example, Tremble and SwissProt. There are also specialized databases that cater to particular research interest. For example, Flybase, HIV sequence databases. In these databases, all the information about HIV virus is stored in structured form, and ribosomal databases, project and database that specialize in a particular organism or particular type of data. Composite databases, It compiles and filters sequence data from primary database. More exhaustive than individual databases include protein sequence data from translated coding region in DNA sequences. For example, OWL and NRDB biosilico. So what are the importance of databases? These all databases which I have defined on the basis of the sources, that is primary databases, secondary databases and composite databases. But there are different type of databases which are depend on natures. So there are two type of databases like sequence databases and structural databases. In sequence databases, all the biological sequences like DNA sequences, RNA sequences and protein sequences are stored. For example, GenBank, DDBJ and ENVM. In structural databases, there are storage of structures of a biological data like DNA, RNA and proteins. So in structural databases, we can get protein structures, gene structures as well from the structural databases. There are some specialized databases used to specific functions. So now come to the point. What are the importance of databases? In terms of biological databases, so databases act as a storehouse of informations from where we can access the data, we can we can analyze the data, we can visualize the data, we can predict the data. For further research, for further analysis, we can use that particular biological data. So databases are used to store and organize data in such a way that information can be retrieved easily. It allows knowledge discovery, which refers to the identification of connections between pieces of information that were not known when the information was first entered. This facilitates the discovery of new biological insights from raw data. Raw data, it means the data which is not processed. Secondary databases have become the molecular biologist reference library over the past decade or so, providing a wealth of information on just about any gene product that has been investigated by the research community. It helps to solve cases where many users want to access the same entire of data, allows the indexing of data. It helps to remove redundancy of data. Here the redundancy of data means the repetition of the data. So I think that you all got these type of informations from these slides, and what are the information of biological data, what is biological databases, what are the importance and how can we use these databases. So there are, like I explained NCBI, I explained DDBJ, GenBank, and there are different type of databases which are included in NCBI, National Center of Biotechnological Informations. From there, if you go that website NCBI, there are drop down menu, all databases, and you can use any type of databases to access sequences and structures from that particular website, that is NCBI. So thank you so much.